Hello everyone, welcome back to my channel. So today we are going to start NCERT audiobook series. तो यहाँ पे हम जो फर्स्ट चैप्टर स्टार्ट करने जा रहे हैं वो है चैप्टर नंबर 1 सेक्सुअल रिप्रोडक्शन इन फ्लावरिंग प्लांट्स. तो स्टार्ट करते हैं. आर वी नॉट लकी दैट प्लांट reproduce sexually? The merits of flower that we enjoy gazing at, the scent and the perfume that we swoon over, the rich color that attract us, are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfness. All all flowering plants show sexual reproduction. A look at the diversity of structure of the inflorescence, flowers and floral parts shows an amazing range of adaptation to ensure formation of the end product of sexual reproduction, the fruit and seeds. In this chapter, let us understand the morphology, structure and process of sexual reproduction in flowering plant angiosperm. The first topic is flower, a fascinating organ of angiosperm. Human beings have had an intimate relationship with the flower since time of immortal. Flowers are object of aesthetic, ornamental, social, religious and cultural value. They have always been used as symbol for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least 5 flowers of ornamental value that are commonly cultivated at homes and in gardens. Find out the names of 5 more flowers that are used in social and cultural celebration in your family. Have you ever heard of floriculture? What does it refers to? To a biologist, flowers are morphological, embryological marvels and site of sexual reproduction. In earlier classes, you have read the various part of flower. Figure 1.1 will help you to recall the part of a typical flower. Can you name the 2 parts in a flower in which the 2 most important unit of sexual reproduction develop? Pre-fertilization, structure and function. Much before the actual flower is seen on a plant, the decision that plant is going to flower has taken place. Several hormonal and structural changes are initiated which leads to differentiation and further development of floral primordium. Inflorescences are formed which bear the floral bud and then the flower. In the flower, the male and female reproductive structure, the androecium and gynoecium, differentiate and develop. You would recollect that the androecium consists of roll of stamen representing the male reproductive organ and the gynoecium represent the female reproductive organ. Stamen, microsporangium, pollen grain. Figure 1.2a shows the 2 parts of typical stamen, the long and slender stalk called filament and the terminal generally bilobed structure called anther. The proximal end of filament is attached to the thalamus or petal of the flower. The number and length of the stamen are variable in flower of different species. If you were to collect a stamen each from 10 flowers, each from different species, or arrange them into a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagram would elucidate the range in shape and attachment of anther in different flowers. A typical angiosperm anther is bilobed with each lobe having 2 theca. It means they are dithecous. Often a longitudinal groove run lengthwise separating the theca. Let us understand the various type of tissue and their organization in the transverse section of an anther. The bilobed nature of an anther is very distinct in the transverse section of anther. The anther is 4-sided tetragonal structure consisting 4 microsporangium located at the corner, 2 in each lobe. The microsporangium further develop and become pollen sacs. They extend longitudinally, although the length of an anther are packed with pollen grains. Structure of microsporangium. In a transverse section, a typical microsporangium appears near the circular in outline. It is generally surrounded by 4 wall layers. Figure 1.3b. The epidermis, endothelium, middle layer and tapetum. The outer 3 wall layer perform the function of protection and help in dehiscence of anther to release pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. Cell of the tapetum possesses dense cytoplasm and generally have more than one nucleus. Can you think how tapetal cell would become binucleated? When the anther is young, a group of compactly arranged homogeneous cells called homogeneous tissue occupies the center of each microsporangium. Microsporogenesis. As the anther developed, the cell of sporogenous tissue undergoes meiotic division to form a microspore tetrad. What would be the ploidy of the cell of the tetrad? As each cell of the sporogenous tissue is capable of giving rise to the microspore tetrad, each one is potential pollen or microspore mother cell. The process of formation of microspore from pollen mother cell PMC through meiosis is called microsporogenesis. The microspores as they are formed are arranged in a cluster of 4 cells, the microspore tetrad. As the anther mature and dehydrate, the microspore dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousand of microspores or pollen grains are formed that are released with the dehiscence of anther. Pollen grain. The pollen grain represent the male gametophyte. If you touch the opened anther of hibiscus or any other flower, you would find deposit of yellowish powdery pollen grain on your fingers. Sprinkle these grains on a drop of water taken on a glass slide under a microscope. You will really be amazed at the variety of architecture, size, shape, color, design seen on the pollen grain from different species. Pollen grain are generally spherical, measuring about 25 to 50 micrometer in diameter. It has a prominent 2-layered wall. The hard outer layer called exine is made up of sporopollenin, which is one of the most resistant organic material known. It can be withstand with a high temperature and strong acid and alkali. No enzyme that degrades sporopollenin is so far known. Pollen grain exine has a prominent aperture which is called germ pore, where the sporopollenin is absent. Pollen grain are well preserved as a fossil because the presence of the sporopollenin. The exine exhibit a fascinating array of pattern and design. Why do you think the exine should be hard? What is the function of germ pore? The inner wall of the pollen grain is called intine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When a pollen grain is mature, it contains 2 layers, the vegetative cell and generative cell. The vegetative cell is bigger, has abundant food reserve and large, irregularly shaped nucleus. The generative cell is small and float in the cytoplasm of the vegetative cell. It is spindle shaped with a dense cytoplasm and a nucleus. In over 60% of the angiosperm, pollen grain are shed at 2-cell stage. In the remaining species, the generative cell divide mitotically to give rise to 2 male gametes before pollen grain are shed 3-celled stage. Pollen grain are species cause severe allergies and bronchial affliction in some people, often leading to chronic respiratory disorder like asthma, bronchitis, etc. It may mention that Parthenium or carrot grass that came into India as a contaminant with important weeds has become ubiquitous in occurrence and cause pollen allergy. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablet as a food supplement. In Western country, a large number of pollen protein form of tablets and syrups are available in market. Pollen consumption has been claimed to increase the performance of athletes and race horses. When once they are shed, pollen grain have to land on the stigma before they lose viability if they have to bring about fertilization. How long do you think pollen grain retain viability? The period of which pollen grain remain viable is highly variable and to some extent depend on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release, and in some members of Rosacea, Leguminosa and Solanaceae they maintain viability for a month. You may have heard of storing semen, sperm of many animals including human for artificial insemination. It is possible to store pollen grain of a large number of species for years in liquid nitrogen of -196 degrees Celsius. Such stored pollen can be used as a pollen bank similar to seed bank in crop breeding programs. The pistil, microsporangium, ovule and embryo sac. The gynoecium represents the female reproductive part of flower. The gynoecium may consist of a single pistil which is monocarpellary or may have more than one pistil, multicarpellary. When there are more than one pistil may be fused together, they called syncarpus, or may be free, they called apocarpus. Each pistil has three parts: the stigma, style and ovary. The stigma serves as a landing platform for the pollen grain. The style is the elongated cylinder part beneath the stigma. The basal bulbed part of the pistil is the ovary. Inside the ovary it is ovarian cavity which is locule. The placenta is located inside the ovarian cavity. Recall the definition and type of placentation that you studied in class 11th. Arising from the placenta are the megasporangium, commonly called ovule. The number of ovule in an ovary may be one: wheat, paddy, mango, or many: papaya, watermelon, orchids. The megasporangium, which is ovule. Let us familiarize ourselves with the structure of typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stall called funicle. The body of ovule fuses with funicle in the region called hilum. Thus, hilum represents the junction between ovule and funicle. Each ovule has one or two protective envelope called integument. Integument encircle the nucellus except at the tip where a small opening called micropyle is organized. Opposite the micropylar end, the chalazal representing the basal part of ovule. Enclosed within the integument is a mass of a cell called nucellus. Cell of nucellus have abundant reserve food material. Located in nucellus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from megaspore. Megasporogenesis. The process of formation of megaspore from megaspore mother cell is called megasporogenesis. Ovule generally differentiates a single megaspore mother cell MMC in a micropylar region of the nucleus. It is a large cell containing dense cytoplasm and prominent nucleus. The MMC undergoes meiotic division. What is the importance of the MMC undergoing meiosis? Meiosis results in the production of 4 megaspore. Female gametophyte. In a majority of flowering plant, one of the megaspore is functional while the other degenerates. Only the functional megaspore develop into the female gametophyte embryo sac. The method of embryo sac formation from a single megaspore is monosporic development. What will the ploidy of the cell of the nucellus MMC, the functional megaspore and female gametophyte? Let us study about the formation of the embryo sac in detail. The nucleus of the functional megaspore divides mitotically to form two nuclei which move to the opposite pole, forming the two nucleate embryo sac. Two more sequential mitotic nuclear division result in the formation of 4 nucleate and later 8 nucleate stage of the embryo sac. It is interesting to note that these meiotic division are strictly free nuclear, that is, nuclear division are not followed immediately by the cell wall formation. After the 8 nucleate stage, cell wall are let down, leading to the organization of typical female gametophyte or embryo sac. Observe the distribution of cell inside the embryo sac. 6 of 8 nuclei are surrounded by cell wall and organized into cell. The remaining 2 nuclei, called pollen nuclei, are situated below the egg apparatus in the large central cell. There is a characteristic distribution of the cell within the embryo sac. 3 cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus is turned consists of 2 synergid and 1 egg cell. The synergid has special cellular thickening at the micropylar tip, which is called as filiform apparatus, which play important role in the guiding the pollen tube into the synergids. 3 cells are at the chelazal end, which is called as antipodal. The large central cell which is mentioned earlier has 2 pollen nuclei. Thus, a typical angiosperm of embryo sac at maturity, though 8 nucleate, is 7 celled. Pollination. In the breeding section, you have learned that the male-female gamete in the flowering plant are produced in a pollen grain and embryo sac respectively. As both gametes are non-motile, they have to be brought together for fertilization to occur. How is this achieved? Pollination is a mechanism to achieve this objective. Transfer of pollen grain shed from the anther to the stigma of a pistil is termed pollination. Flowering plant has evolved as amazing array of adaptation to achieve pollination. They make use of external agent to achieve pollination. Can you list possible external agent? Kind of pollination. Depending on the source of pollen, pollination can be divided into 3 types. First, autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grain from anther to stigma of the same flower. In a normal flower which open and exposes the anther and the stigma, complete autogamy is rare. Autogamy in such flower requires synchrony in pollen releases and stigma respectively, and also the anther and the stigma should lie close to each other so that the self-pollination can occur. Some plants such as viola, common pansy, oxalis and commelina produce 2 types of flower. Chasmogamous, flower which are similar to flower of same species which exposed anther and stigma, and clistogamous flower which do not open at all. In such flower, the anther and stigma lie close to each other. When anther dehiscence in the flower bud, pollen grain come in contact with the stigma to effect pollination. Thus, clistogamous flowers are invariably autogamous as there is no chance of cross-pollination landing on the stigma. Clistogamous flowers produce assured seed sets even in the absence of pollinator. Do you think the clistogamy is advantage or disadvantageous in the flowering plant? Why? Gymnogamy. Transfer of pollen grain from the anther to the stigma of another flower of the same plant. Although gymnogamy is functionally crossed pollination involving a pollinating agent, genetically it is similar to autogamy since the pollen grain come from same plant. Genogamy, transfer of pollen grain from anther to the stigma of different plant. This is the only type of pollination which during pollination brings genetically different type of pollen grain to the stigma. Agent of pollination. Plants use 2 abiotic, wind and water, 1 biotic, which is animal agent to achieve pollination. Majority of plant use biotic agent for pollination. Only a small proportion of plant use abiotic agent. Pollen grain coming in contact with the stigma is a chance factor in both wind and water pollination. To compensate for these uncertainties and associated loss of pollen grain, the flower produce enormous amount of pollen when compared to the number of ovules available for the pollination. Pollination by wind is most commonly among abiotic pollination. Wind pollination are required that the pollen grain are light and non-sticky so that they can be transported in wind current. They often processes well exposed stamen so that the pollen are easily disappeared into the wind current, and large often feathery stigma to easily trap airborne pollen grains. Wind pollinated flowers often have single ovule in each ovary and numerous flowers packed into the inflorescence. A familiar example is corn cob. The tassel you have seen are nothing but the stigma and style which wave in the wind to trap pollen grain. Wind pollination is quite common in grasses. Pollination by wind is a quite rare in flowering plant and is limited to about 30 genera, mostly monocotyledons. As against this, you would recall that water is regular mode of transport for the male gamete. Among the lower plant groups such as algae, bryophyte, tetraidophyte, it is believed particularly for some bryophytes and tetraidophytes that their distribution is limited because of the need of water for the transport of male gamete fertilization. Some example of water pollinated are Vallisneria hydrilla, which grow in fresh water, and several marine seagrasses such as Zostera. Not all aquatic plants use water for pollination. In majority of aquatic plants such as water hyacinth and water lily, the flower emerges above the level of water and pollinated by insect or wind, as the most of the land plant. In Vallisneria, the female flower reach the surface of water by the long stalk, and male flower of pollen grain are released onto the surface of water. They are carried passively by the water currents. Some of them eventually reach the female flower and the stigma. In another group of water pollinated plants such as seagrasses, female flower remain submerged in the water and pollen grain are released inside the water. Pollen grain in many species are long ribbon like, and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. In most of the water pollinated species, pollen grain are protected from wetting by mucilaginous covering. Both wind and water pollinated flower are not very colorful and do not produce nectar. What would be the reason of this? Majority of flowering plants use a range of animal as pollinating agent. Bees, butterflies, beetle, wasp, ant, moth, birds, sunbird and hummingbird and bat are common pollinating agent. Among the animal, insect, particularly bees, are the dominant biotic agent. Even larger animals such as primates, lemur, herbivolid, tree-dwelling rodent or even reptiles, gecko, lizard and garden lizard have also been reported as a pollinator in some species. Often flowers of animal pollinated plants are especially adapted for the particular species of animal. Majority of insects pollinated flowers are large, colorful, fragrant and rich in nectar. When the flowers are small, a large number of flowers are clustered into an inflorescence to make them conspicuous. Animals are attracted to the flower by colorful and or fragrance. The flowers pollinated by flies and beetles secrete foul odor to attract these animals. To sustain animal visit, the flowers have provide reward to the animal. Nectar and pollen grain are the unusual floral reward. For harvesting the reward from the flowers, the animal visitor comes in contact with the anther and stigma. The body of the animal get a coating of pollen grain, which are generally sticky in animal pollinated flower. When the animal carrying pollen on its body come in contact with a stigma, it brings pollination. In some species, the floral reward are providing to the safe place to lay egg. In an example, it is that of the tallest flower of Amorphilus. The flower itself is about 6 feet in height. A similar relationship exists between species of moth and a plant yucca, where both species moth and the plant cannot complete their life cycle without each other. The moth deposits its egg in the locule of the ovary, and flower in turn get pollinated by moth. The larva of the moth came out of the egg as the seed start developing. Why don't you observe some flower of the flowering plant or any other available to you? Cucumber, mango, people, coriander, papaya, onion, lobia, cotton, tobacco, rose, lemon, eucalyptus, banana. Try to find out these which animal visit them and whether they could be pollinated. You have to patiently observe the flower over a few days and a different time of days. You could also try to see whether there is any correlation in characteristic of flower to the animal that visited. Carefully observe it. Any of the visitor come in contact with the anther of the stigma, as only such visitor can bring about pollination. Many insects may consume pollen or the nectar without bringing about the pollination. Such floral visitors are referred to a pollen nectar robber. You may or may not be able to identify the pollinator, but you will surely enjoy your effort. Outbreeding devices. Majority of flowering plant produces hermaphrodite. Flower and pollen grain are likely come in contact with the stigma of the same flower, continue to self-pollination, result in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and encourage cross-pollination. In some species, pollen grain releases stigma and respectively are not synchronized. Either the pollen is released before the stigma become respective or stigma become respective much before the release of pollen. In some other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of same flower. Both these devices prevent the autogamy. The third device to prevent inbreeding is self-incompatibility. This is genetic mechanism and prevents self-pollination from the same flower or other flower of the same plant from fertilizing the ovule of inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is the production of unisexual flower. In both male and female flower are present on the same plant, such as castor and maize, monoecious. It prevents autogamy but not geitonogamy. In several species such as papaya, male and female flower are present on different plant. That is, each plant is either male or female dioecious. This condition prevents both autogamy and geitonogamy. Pollen pistil interaction. Pollination does not guarantee the transfer of the right type of pollen. Compatible pollen of the same species as stigma. Often pollen of the wrong type from either from each species or from the same plant. If it is self-compatible, also land on the stigma. The pistil has ability to recognize the pollen whether it is right type, compatible, or wrong type, incompatible. If it is right type, the pistil accept the pollen and promote the post-pollination event that leads to fertilization. If the pollen is wrong type, the pistil reject the pollen by preventing the pollen germination on the stigma or pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of continuous dialogue between pollen grain and the pistil. This dialogue is mediated by the chemical component of the pollen interacting with those of the pistil. It is the only in the recent year that botanists have been able to identify some of the pollen and pistil component and the interaction leading to the recognition followed by the acceptance or rejection. As mentioned earlier, followed compatible pollination, the pollen grain germinates on the stigma to produce pollen tube through one of the jump pore. The content of the pollen grain move to the pollen tube. Pollen tube grow through the tissue of a stigma and style reaches to the ovary. You would recall that in some plant, pollen grain are shed at two-celled condition, a vegetative and generative cell. In plant, the generative cell divides and forms the two male gamete during the growth of pollen tube in the stigma. In plant which shed pollen in three-celled condition, pollen tube carries the two male gamete from the beginning. Pollen tube, after reaching the ovary, enter the ovule through the micropyle micropilar and then enters one synergid through the filiform apparatus. Many recent studies have been shown that filiform apparatus present in the micropilar part of the synergid guides the entry of pollen tube. All these events from the pollen deposition of the stigma until pollen tube enters as a ovule are observed referred to as a pollen pistil interaction. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of pollen. The knowledge gained in this area would help the plant breeder in the manipulating pollen pistil interaction even in incompatible pollination to get desired hybrid. You can easily study pollen germination by dusting some pollen from the flower such as pea, chicken pea, crotalaria, balsam, vinca on a glass slide containing a drop of sugar solution about 10%. After about 15 to 30 minutes, observe the slide under the low power lens of the microscope. You are likely to see pollen tube coming out of the pollen grain. A breeder is interested in crossing different species and often genera to combine desirable character to produce commercially superior varieties. Artificial hybridization is one of the major approach for crop improvement program. In such crossing experiment, it is important to make sure that only the desired pollen grain are used for pollination and stigma is protected from the contamination from unwanted pollen. This is achieved by emasculation and bagging technique. If the female parent bear bisexual flower, removal of anther from the flower bud before the anther dehiscence using a pair of forceps is necessary. This step is referred to as emasculation. Emasculated flower have to be covered with a bag of switchable sides, generally made up of butter paper, to prevent contamination of its stigma with unwanted flower. This process is called bagging. When the stigma of bagged flower attain respectivity, mature pollen grain collected from the anther of the male parent are dusted on the stigma and the flowers are rebagged and the fruit allowed to develop. If the male parent produce unisexual flower, there is no need for the emasculation. The female flower bud are bagged before the flower open. When the stigma became respective, pollination is carried out using the desired pollen and the flower rebagged. Double fertilization. After entering one of the synergid, the pollen tube releases the two male gametes into the cytoplasm of the synergid. One of the male gametes moves toward the egg and fuses with its nucleus, thus completing the syngamy. This results in the formation of diploid cell, the zygote. The other male gamete moves toward the two polar nuclei located in the central cell and fuses them with to produce a triploid primary endosperm nucleus span. As this involves a fusion of three haploid nuclei, it termed as a triple fusion. Since two types of fusion, syngamy and triple fusion, take place in the embryo sac, the phenomena is formed as which is called as double fertilization, an event unique to the flowering plant. The central cell after the triple fusion become the primary endosperm cell PEN and develop into the endosperm while the zygote develop in the embryo. Post fertilization structure and events. Flowering double fertilization. Following double fertilization, events of the endosperm and embryo development, maturation of ovule into seed and ovary into fruit are collectively termed as post-fertilization event. Endosperm development precedes embryo development. Why? The primary endosperm cells divides repeatedly and forms a triploid endosperm tissue. The cell of this tissue are filled with a reserved food material and are used for the nutrition. The developing embryo. In the most common type of endosperm developing, the PEN undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occur and the endosperm becomes cellular. The number of free nuclei formed for cellularization varies greatly. The coconut water from the tender coconut that you have familiar with is nothing but a free nuclear endosperm made up of thousands of nuclei and are surrounded by a white kernel in the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, example pea groundnut beans, before seed maturation, or it may persist in the mature seeds, example castor and coconut, and be used up during the seed germination. Spread open some seed of castor, pea, bean, groundnut, fruit of coconut, and look for the endosperm in each case. Find out whether the endosperm is persistent in cereals, wheat, rice, and maize. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygote divide only after the certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Through the seed differ greatly. The early stage of embryo development, embryogeny, are similar in both monocotyledons and dicotyledons. Depict the stage of embryogeny in a dicotyledonous embryo. The zygote give rise to the proembryo and subsequently to the globular heart-shaped and mature embryo. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledons is epicotyledons, which terminate with the plumule or a stem tip. The cylindrical portion below the level of cotyledons is hypocotyle, that terminate at its lower end of the radicle and tip root. The root tip is covered with a root cap. Embryo of monocotyledonous possesses only one cotyledon. In the grass family, the cotyledon is called scutellum, that is situated toward one side lateral and embryonal axis at its lower end. The embryonal axis has the radicle and root cap enclosed in an undifferentiated sheath which is called coleorrhea. The portion of the embryonal axis is above the level of attachment of scutellum in the epicotyle. Epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure, the coleoptile. Soak a few seed in water, say of wheat, maize, pea, chickpea, groundnut overnight. Then split the seed and observe the various part of embryo and the seed. In an angiosperm, the seed is final product of asexual reproduction. It is often described as a fertilized ovule. Seed are formed inside fruit. A seed typically consists of seed coat, cotyledons, and the embryo axis. The cotyledons of the embryo are simple structures, generally thick and solvent due to the storage of food reserve as legumes. Mature seed may be non-albinous and ex-albinous. Non-albinous seed have no residual endosperm as it is completely consumed during embryo development. It means pea, groundnut. Albinous seed retain a part of endosperm as it not completely used up during embryo development. Example wheat, maize, barley, castor. Occasionally in some seed such as black pepper and beet, remnant of the nucleus are also persistent. The residual persistent nucleus is the perisperm. Integument of ovule harden as tough protective seed coat. The micropylar remain as a small pore in the seed coat. This facilitates entry of oxygen and water in the seed during germination. As the seed mature, its water content is residue and seed become relatively dry, 10 to 15% moisture by mass. The general metabolic activity of the embryo slow down. The embryo may enter a state inactivity called dormancy, or if favorable conditions are available, adequate moisture, oxygen and suitable temperature, they germinate. As ovule mature into seeds, the ovary develop into a fruit. It means the transformation of ovule into seed and ovary into fruit proceeds simultaneously. The wall of the ovary develop into the wall of the fruit called pericarp. The fruit may be freshly as in guava, orange, mango, etc., or may be dry as groundnut and mustard, etc. Many fruit have involved in dispersal mechanism that you have studied in an earlier classes. Is there any relationship between number of ovule in an ovary and number of seed present in a fruit? In most plant, by the time of the fruit develop from the ovary, other floral part degenerates and fall up. However, in a few species such as apple, strawberry, cashew, etc., the thalamus also contribute to fruit formation. Such fruit called false fruit. Most fruit, however, develop only from the ovary and are called as true fruit. Although in most of the species fruit are the result of fertilization, there are few species in which fruit develop without fertilization, which is called as a parthenocarpic fruit. Banana is one of them example. Parthenocarpy can be induced through the application of growth hormone, and such fruit are seedless. Seed offer several disadvantages of angiosperm. Firstly, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seed have been better adaptive strategies for dispersal of new habitat and help in the species to colonize in other areas. As they have sufficient food they have sufficient food reserve, young seedlings are nourished until they are capable of photosynthesis of their own. The hard seed coat provide protection of the young embryo. Being products of sexual reproduction, they generate new genetic combination leading to variation. Seed is the basis of our agriculture. Dehydration and dormancy of the mature seed are crucial for storage of seed, which can be used for food throughout the year, also to raise crop in the next season. Can you imagine agriculture is the absence of seed or in the presence of seed, which germinate straight away soon after formation and cannot be stored? How long do the seed remain alive after they disappeared? This period again varies greatly. In a few species, the seed lose viability within a few months. Seed of a large number of species of the several years. Some seed can remain alive for hundreds of years. There are several records of very old yet viable seed. The oldest is that of Lupinus arcticus excavatus from Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. A recent record 2,000-year-old viable seed is of the date palm Phoenix Dactylophora, discovered during the archaeological excavation at King Herod's place near Dead Sea. After completing a brief account of sexual reproduction of flowering plant, it would be worth attempting to comprehend the enormous reproductive compatibility of same flowering plant by asking the flowering question: how many eggs are present in an embryo sac? How many embryo sac are present in an ovule? How many ovule are present in an ovary? How many ovaries are present in a typical flower? How many flowers are present on a tree? And so on. Can you think of some plant in which fruit contain very large number of seed? Orchid fruit are one such category, and each fruit contains thousands of tiny seeds. Similar in the case in fruit of some parasitic species such as Orobanche and Stiga. Have you seen a tiny seed of ficus? How large is the tree of ficus developed from the tiny seed? How many billion of seed does from each ficus tree have? Can you imagine any other example in which such tiny structure can produce such large biomass over the year? Mixes and polyembryony. Although seeds in general are product of fertilization, a few flowering plants such as some species of Asteraceae and grasses have evolved a special mechanism to produce seed without fertilization, which is called as apomixis. What is fruit production without fertilization called? Thus, apomixis is formed of asexual reproduction that mimics sexual reproduction. There are several ways of development of apomictic seed. In some species, the diploid egg is formed without reduction division and develops into the embryo without fertilization, more often as in many citrus and mango. Varieties of some nuclear cell surrounded by the embryo sac start dividing, protrude into the embryo sac and develop into embryo. In such species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is referred to as polyembryony. Take out some seed of orange and squeeze them. Observe many embryo of different size, shape from each seed. Count the number of embryos in each seed. What would be the genetic nature of apomictic embryo? Can they be called clones? Hybrid variety of several of our food and vegetable crop are being extensively cultivated. Cultivation of hybrid had been tremendously increased productivity. One of the problem of hybrid is that hybrid seed have to be produced every year. If the seed collected from the hybrid are sown, the plant is in the progeny will segregate and do not maintain hybrid characters. Production of hybrid seed is costly and hence the cost of hybrid seed becomes too expensive for the farmers. If these hybrids are made into the apomix, there is no segregation of characters in the hybrid progeny. Then the farmer can keep on using the hybrid seed to raise new crop year after year, and he does not have to buy hybrid seed every year. Because of the importance of apomixis in hybrid seed industries, active research is going on in many laboratories and around the world. Understand the genetic of apomixis and to transfer apomictic gene into the hybrid varieties. Summary: flowers are seat of sexual reproduction in angiosperm. In flower, androsium consists of stamen represent male reproductive organ, and gynoecium consists of pistil represent the female reproductive organ. A typical anther is bilobed, dithecous, and tetraporagnate. Pollen grain develop inside the microsporangium, four-walled layer, the epidermis, endothecium and middle layer, and the tapetum, surrounded by the microsporangium. Cell of the sporogenous tissue lying in the center of the microsporangium undergo meiosis, microsporogenesis to form tetrad of microspore. Individual microspore mature into pollen grain. Pollen grain represent the male gametophyte generation. The pollen grain have two-layered wall, the outer exine and inner antine. The exine is made up of sporopollenin and has germ pole. Pollen grain may have two cell, vegetative cell and generative cell, of three cell, a vegetative cell and two male gamete. At the time of shedding, the pistil has three-part stigma, style and ovary. Ovule are present in the ovary. The ovule have stalk called funicle, protective integument and an opening called micropyle. The central tissue is the nucellus in which the archiporium differentiates. A cell of the archiporium, the megaspore mother cell, divide mitotically and one of the megaspore form the embryo sac. The female gametophyte, the mature embryo sac shed at 7 cell and 8 nucleate at micropylar and is the egg apparatus consisting of two synergids and an egg center and egg cell. As the chelazal end are three antipodals, at the center is a large central cell with the two polar nuclei. Pollination is the mechanism of transfer of pollen grain from anther to stigma. Pollinating agent are either abiotic, wind and water, or biotic animals. Pollen pistil interaction involve all events from the landing of pollen grain on the stigma until the pollen tube enters the embryo sac when the pollen is compatible, or pollen inhabitation when the pollen is incompatible. Following compatible pollen grain germinate on the stigma and resulting the pollen tube grow through the style, enter the ovule and finally discharge the two male gamete in one of the synergid. Angiosperm exhibit double fertilization because two fusion events occur in embryo sac, namely syngamy and triple fusion. The product of these fusion are diploid zygote and triploid. Primary endosperm nucleus in the primary endosperm cell, zygote develop in the embryo, and primary endosperm cell form the endosperm tissue. Formation of endosperm always proceed development of embryo. The developing embryo possesses through different stages such as pro-embryo, globular and heart-shaped stage. Before maturation, mature dicotyledonous embryo has two cotyledons and an embryonal axis with epicotyle and hypocotyle. Embryo of monocotyledons have a single cotyledon. After fertilization, ovary develop into fruit and ovule develop into seed. The phenomena called apomixis is found in some angiosperm, particularly in grasses. It results in the formation of seed without fertilization. Apomix have several advantages in horticulture and agriculture. Some angiosperm produces more than one embryo in the seed. This phenomena is called polyembryony. Thank you so much for watching. अगर आपने full video देखी है so heads off to you. आपने बहुत अच्छा काम किया है. You have read it full chapter in very less time, and do like, share and subscribe my channel and also share to others. थैंक यू सो मच. एंड आगे की वीडियो भी ज़रूर देखिएगा. थैंक यू.